Hello, good morning, and welcome to our last seminar of this spring series. Thank you for joining, and thank you to Dr. Valerio Hoyos Villegas, who is joining us from Canada, from McGill University. Valerio is originally from Colombia. He got his master's in crop physiology from the University of Missouri in Columbia, and then a PhD from Michigan State University in plant breeding, genetics, and biotechnology. Since 2019, he's been an assistant professor in plant breeding and genetics at McGill University in Canada, where he holds teaching and research responsibilities. He teaches undergrad statistics and graduate level plant breeding and genetics. As you can guess from the video that you just saw, Valerio's area of expertise is breeding and genetics of pulses, and also developing phenomic tools for the improvement of complex traits. Valerio is an editor of several journals and also has served in leadership roles in the National Association of Plant Breeding and the North American Plant Breeding Phenotyping Network. Because he has experimented some problems with Zoom, Valerio has pre-recorded his presentation, although he is with us. I'm gonna stop sharing here and give the floor to Brandon, who is gonna show Valerio's presentation, and then Valerio is here, he's gonna answer questions at the end. Please post your questions in the chat, as always. And again, thank you for joining, and I'm gonna stop sharing here.
Hello, everybody, and thank you for the invitation to speak. My name is Valerio Hoyos Villegas. I am an assistant professor of plant breeding and genetics at McGill University. And today my talk is titled Phenomics-Enabled Breeding, Crop Improvement for Solving Grand Challenges. So the general question is that in terms of agricultural gains, are we closing the yield gap? And so in 2010, the projection is that as of 2010, the projection was that the world food demand would require 1.25% increase per annum. Some of the ways that we can measure this is this yield gap, which is the difference between the potential yield and the on-farm yield, or the percent of the farm yield. One of the major conclusions is that except for a few crops, most crops are behind. This is the number of food insecure people in the world has increased, and has increased over the years. And this factor or situation has been dramatically worsened by issues with supply chain, logistics, and distribution around the world due to the COVID-19 pandemic. So this puts further pressure on the local food security, as well as food trade between countries. So the breeding cycle is essentially comprised of three. We can summarize it in 3 words. One of them is intermate. The second one is evaluate. The third one is select, and then repeat. We can frame this concept around the breeder's equation, or the genetic gain equation, which is essentially the gain over time, selection intensity, the selection accuracy, the additive or most useful component of the genetic variance, which is this additive standard deviation, and the number of years it takes to complete one cycle. Breeders have come up with a number of ways of modifying all components, or most of the components in this equation. But one of the ones that remains a bottleneck is phenotyping, and particularly phenotyping as it pertains to increasing genetic variance, or maintaining genetic variance throughout time, while thus rendering a breeding program more sustainable in terms of its sustained progress. So this additive component, additive standard deviation, can be approximated by a linear model, where we have a phenotype, a grand mean, a genotype, an environment, an interaction of the two, and within error. An old versus new concept is that in relation to this is that the continuous variation or quantitative variation is modeled as the joint effects of multiple Mendelian loci. However, we do know through breeding efforts and genetics efforts that the effects indeed are small and the QTL are numerous. Okay, and this has been shown in a number of other fine mapping experiments where very, very large mapping populations have only resulted in the induction of very low recombination rates. So in relation to this, we want to approximate this response variable and this response variable, which is a phenotype, through the genotype, which is essentially the genotypic and not the breeding value itself, right? And secondly, we want to make we can do this by making SNP-based predictions. Okay. We have a number of tools such as GBLUP or Ridge Regression BLUP, which are equivalent when large numbers of QTL are involved, or no major QTL are involved and they're evenly distributed. But we know that this might not necessarily be the case, but it is one of the ways we've made progress on this. So essentially, we want to reduce this latter three components of the model to as close as zero as possible. And one of the ways that we are making progress on this is by having more and more accurate phenotyping. But in the future, we can the phenotyping data that we're gathering will likely become also a powerful tool to conduct predictive phenotyping. Okay, so utilize historical phenotype data to. What I mean by that is that in relation also to an old concept is that we have the multiple loci are also associated with all the environmental effects. But we know that this is way more complicated than it was first thought, where it is often required to incorporate biophysical process models in order to better understand the environmental effects, or these three latter components of this model, and how these biophysical processes are influenced by the resource use, or rather the interaction between the genotype and the environment, and in relation to our trait of interest, which is the genotypic value. So in essence, one of the ways that we've approached estimating the genotypic value is through mapping QTL, and many of you are familiar with a figure like this, where we have a chromosomal or genomic position or chromosomal position, and we have usually a threshold which enables us to detect a location that is significant. So we can say, for example, that this interval could be responsible, despite having areas of low and high significance or QTL association. It would be responsible for the quantitative variation of yield, for example. But this yield variable can be decomposed into various components. In the case I work with pulses, so in the case of beans, we could say that the yield could be also comprised by numerous individual QTL, which are made up of, say, seeds per pod, which is a positive trait, number of pods, which is also a positive trait, positive selection trait, and seed size, which can be positive or negative in our case, as well as pod abortion rate, for example. So these, as you can see, some of these traits might be above the threshold, and other ones can be below the threshold, depending on the power of our yield of our QTL study. So what we suggest is that instead of trying to find means of selecting for a single or multiple yield QTL, which might have variable responses in relation to their individual components, what we take that same trait, we try to find these individual component QTLs, and we develop one trait that is based on high throughput phenotyping approaches. So we're now reaching the potential of doing this by the myriad of sensors that are available to us scientists now. So as a sort of the the background behind this and this how it is integrates into technology that we are developing here at McGill is essentially the what we call the Eastern Canadian Plant Phenotyping Platform, or ECP3. And this is a number of initiatives, but one of them is the construction of a field phenotyping vehicle, which we have just now in the process of completing, and will be deploying fully, not just at an experimental rate, this year. Okay, and so this system can perform things such as morphometric imaging, for example, for the reconstruction of canopy architectures, LiDAR for volume and height, this RGB light and visible imaging for the detection of, say, disease lesions and disease development. We can do chlorophyll fluorescence kinetics for photosynthetic efficiency processes, as well as thermal, hyperspec, and multispec imaging. As well, all these sensors are all RTK, have all incorporated RTK navigation, as well as weather sensors at the microenvironment level, rather. So that these are sensors that allow us to also decompose environmental data at the plot level or plant to plant level. That leads me to talk about a second area of research, which is affordable phenomics. And I just want to mention a couple of areas here. One of them is a case study in developing sensors for the detection of diseases using wearable sensors, and this is in collaboration with the University of Glasgow. And then a second case study is the biological nitrogen fixation in soybean with using hyperspectral imaging, and this is a collaboration with the University of Waikato Ag Research and Lincoln Agritech in New Zealand. In terms of how we came about using bendable pH sensors for phenotyping, we understand very well that the white mold is a disease that is pH dependent. It's a devastating disease, but its development is quite linked to changes in cellular pH. This is a process that the fungal pathogen requires in order to develop its infection. It's a necrotrophic pathogen, therefore it needs to decrease pH to kill the cells surrounding it so that it could further infect other tissues. And this is mainly conducted through the exudation of oxalic acid. So that lowers pH, and this enables further infection. So we started this collaboration in a small way by measuring simply pH of apoplastic fluid, and we knew that the wearable potential metric pH sensors were something that was in the horizon in areas of electronic engineering and or wearable sensor engineering. And so we wondered, can we use this to monitor pH in plants and detect whether changes at the localized pH level or enable us to track development of disease, or how does that affect overall plant homeostasis? So the difference was to understand this these changes. So we used the typical straw test, which is a system that allows us to inoculate and then rate varieties based on their resistance. And we had a number of varieties listed there that range from, there's no complete resistance in white mold, so they range from intermediate tolerant, moderately tolerant, and susceptible. And then our sensor setup, we had the sensitive electrode system that was connected to an Arduino platform, and we simply put the attach the sensor system setup on the around and on around the site of infection. But essentially, our experiment suggested that after inoculation, pH did change with our standard pH meter measurements, and as well as in the mock inoculated treatment and in the inoculated treatment, as well in some of them in the inoculated treatment, there were slight decreases in pH. The major trend or pattern that we observed was that in there were all the blue lines on top there show that the inoculated partially resistant varieties always increased, or there was a major change in voltage always in the same direction, regardless of in the inoculated and mock inoculated. And then the orange lines, there was consistent pH voltage decrease, which is calibrated to a particular pH change. And this pattern was found to be consistent between under both mock inoculated and inoculated treatment. So the summary on this is that across mock inoculated and inoculated treatments, cultivars showed consistent pH and voltage readings. So we were able to, in fact, to track these changes in pH. And the largest shifts in pH were observed in the susceptible cultivars. We did also find a weak correlation between the voltage readings and the straw test scores. So these preliminary results have led us to the TESLA project, which is a multi-institution project and aimed at the development of electronic, what we call transient electronics for sustainable digital agriculture, or TESLA. Second case study that I want to mention was BNF, that's interest in tracking biological nitrogen fixation in soybeans and pulses. And as many of you know, that BNF is a process through which legumes form symbiosis with soil bacteria, and they convert atmospheric nitrogen into usable forms of nitrogen for the plant. So the objective was to develop a quick, cheap, and non-destructive and portable way of making BNF measurements as much as possible. And for this, we took two approaches. One of them was a spectral approach, and the other one was a hyperspectral approach. The first one, we used Raman shift, or Raman spectroscopy. And the second one, we used hyperspectral imaging. So we ran two sets of experiments. One of them was no BNF, where there was no rhizobium present and inorganic nitrogen. And the expected range was that as we increase nitrogen concentration, inorganic nitrogen concentration, then we had total nitrogen we were expected to increase. And the alternate situation was that we had plus BNF treatment, where we regulated the amount of rhizobium activity by adding both rhizobium and inorganic nitrogen at various concentrations, and we regulate and we reduce or rather throttled the level of activity of rhizobium and therefore fixation due to this inhibitive effect. So indeed, we had some questions, particularly around the second scenario, where if is there any reference between these treatments, rather these differences, these various nitrogen concentration treatments, can we pick apart, despite the fact that total nitrogen in the plant was fairly was expected to be uniform? Is there any difference that we can pick up under Raman spectra? If no, then the method is not sensitive to BNF. If yes, then are they the same for the no rhizobium set? If that is so, then there is there must be some nitrogen related stresses that still exist in the pattern, and but therefore our experiments should be revised. But if no, then we do indeed have in our hands the means for detecting BNF specific signals. So this was indeed what we observed. We were very excited to pick up these expected signals, where total BNF at variable levels of BNF rates where the maintained total nitrogen, but the observed BNF rate did actually increase as the treatments changed. In terms of models that we could develop based on this experimental setup, we used Raman spectroscopy. Did indeed detect significant changes or significant linear relationship between the various treatments, and so this has led us to go into other patterns. And as expected, in the minus BNF, we found no relationship. So this confirms that we have a BNF specific signal operating here. The second method was hyperspectral imaging, and in this case, what we wanted to do is to determine whether we could conduct analysis and modeling of hyperspectral imaging data cubes using the rhizobia detection model, and then a nitrogen, and then a series of models, the rhizobia detection model, the nitrogen exposure model, and then the N15 estimation model. So this is the process that we took. We have the input data, which are simply images from our leaves at the various stages, and then we extracted foreground. We went through the entire pipeline, and at the end, what we were interested in was detecting through thresholding and confidence thresholding. We wanted to know which, just based on pixel classification, what was the whether the model would actually classify as a rhizobia detection or a nitrogen detection. So which treatment did it belong to? And so with when we took a couple of approaches here, quantitative results, but essentially we're interested mostly in the accuracy and precision of the model. So that is, if we, regardless of whether we went to a pixel-wise approach or a leaf-wise approach, our accuracy and precision, we're still fairly reasonable. They are still have some means for improvement. But the results are promising, meaning that there is a signal there that we can actually enhance. So essentially, in conclusion to my talk, the genotype-phenotype link is actually gaining better bandwidth. We are maintaining, and the linkage is much more, we're much more capable of enhancing this linkage now with field-based phenotyping and our ability to assess changes in phenotypes in the field. And our models exclusively detected BNF patterns, and so we need to continue working on these types of calibrations. Vibrational spectroscopy, such as Raman, is able to pick differential chemical differences due to specific compounds, and we're yet to interrogate which compounds these might be, and these will enable us to develop better models in the future. And hyperspectral imaging has some promise in with models with independent signals validated, such as N15 and these plus-minus BNF systems. So I just want to acknowledge my lab, members of my lab, grad students and technicians and undergraduate students, as well as all of our collaborators in the US, New Zealand, and here in Canada, as well as our funding agency. Any questions? You feel free to reach me at my email or on Twitter. The last thing, if you're a student or postdoc interested in pulse breeding and phenomics, feel free to contact me.
Much. As I said, Valerio is present and ready to answer questions. Very interesting, Valerio. A question got to me. What type of, when you do this image evaluation, is it a plot-based or individual plant-based?
Image of, yeah, are you referring to the
biological nitrogen fixation?
Oh, and BNF. Well, the BNF project is still very much at the rather proof of concept stage. We're still transitioning to, we're just starting to design and run the first soil experiments and transition into field experiments this year. Yeah. So, but so that data was under, let's say, highly controlled conditions, particularly from the soil perspective, that were so that we could regulate the amount of BNF activity.
Okay. Good. Any questions? You can post them in the chat, or you can just open the microphone and ask them to. I'm checking if there's anybody with a raised hand. So the BNF, which species did you use for that, Valerio?
For which one? For BNF?
Yes, for BNF.
We use soybeans and common bean.
Soybeans and?
Common bean and Phaseolus, yeah. Mm-hmm.
Okay. Okay. A question here from Tia. What do you think are the main constraints for wide adoption of HTP in plant breeding?
Well, that's a good question. Just like, you know, anybody that runs a breeding program will know that breeding, in addition to the science, is also largely also a resource management exercise, right? So where you deploy your efforts and your resources can effectively dictate what your rate of gain might be, right? So, and given the long-term nature of breeding, you know, and um, uh, the fact that any potential, you know, disruption to that rate of change might compromise the rate of genetic gain of the program, it's very understandable how, you know, breeders, we can be we're very that, you know, there's got to be very high levels of validation and confirmation in order to adopt new technologies, and this has happened with many and I don't really see this as a weakness or a deficiency in any way. I think it's a very appropriate way of thinking about with the adoption of a new technology, because in a way will dictate in many ways it will dictate the direction of your program, right? So that be that with high throughput phenotyping or genomic selection, et cetera, that just needs to be carefully considered. And this is, you know, that this has been the debate throughout plant breeding research for a very long time. But adoption, you know, it's all very tailored, right? At this point, you know, there's a wide world of possibilities in terms of high throughput phenotyping, and that's going to be, it's evolving in the sense that every program, depending on the species and the needs and the goals, will almost need to develop tools that will enable them to perform selection for the characteristics that they want to improve. And so that is very much a budding area of research.
I have a question here from Raja. It says, thanks, Valerio. Could you please share the equipment you're using for HPI and the investment cost estimators? Is there a publicly available information or?
Well, it's not public, but so this is why I'm a big advocate for affordable phenomics. Okay, that's why I included that in the talk, and that is because generally, as you, maybe that's what you're alluding with your question, is that um, for example, the equipment behind me is sort of towards the high end of the price range. Okay, where the platform, after everything is said and done, costs in the range of millions of dollars, and you know, not every breeding program is able to adopt them. Okay, not that's not taking into account the needs or the skill set necessary to process the data, right? So affordable phenomics is an area that I think will need to really gain a lot of momentum in order for it to be adopted as well by breeding programs. So that's that. I work sort of on that range of the spectrum, but also on the affordable phenomics range of the spectrum, where, for example, you know, the pH sensors are an idea that at the moment they cost below something like less than $7 per sensor, right? And so you can deploy hundreds of them or however many you want if you're using in the greenhouse or in the field. And that makes it a bit more affordable if provided we develop sufficiently reliable tool. Same thing with BNF, for example, where you know, a Raman spectrometer is somewhere in the range of 17,000, and that's a one-off purchase, which as long as you maintain your model calibrated, it's going to be useful. The so I don't know. There's a question there, Carlos, by Raja as well. This is the cost.
Yeah.
But yeah, so I don't know if that answers your question, Raja.
So the other, okay, thank you. The other question we have there is, what is the average heritability value for phenomic traits?
So we try to focus on traits that are low heritability. So a big this is sort of the overall message that I was trying to deliver with the talk, was that we try to focus and develop systems that are applicable for low heritability traits. Okay, so for example, with the phenotyper, we use morphometric imaging. Have a project collaboration with bioresource engineering here at McGill, where we're trying to reconstruct canopy architecture of chickpea plants. Okay, and and that's generally a low heritability trait. And yeah, but we use those traits as the end goal, but in the process of validation, we might include high heritability traits also to verify that we are picking up on genetic signals that are well understood. Right. So in the case of beans, for example, there are major genes that determine seed coat coloring and modifications of seed coat composition and coloring. And there's those are major genes that have been mapped using GWAS. And so we use those as means of validation. But ultimately, the purpose, I think, this is I think this would be the ultimate goal in the breeding committee, would be to implement these as the means for increasing accuracy for selection of low heritability traits.
So let's say you are successful in developing affordable high throughput phenotyping. There's two questions. One is, what will become the next bottleneck in the breeding equation? What do you think? And then the second one is, sometimes, and this was my experience when I worked with Syngenta, it was not an issue, the image acquisition. The issue was getting the information back in time for making a decision for the breeders to do the intercrossing before flowering, for example, if you want to maximize your genetic gain. So two questions, different, but.
So the first question is, what's going to be the next bottleneck, right? So, well, I don't think we're through with the first bottleneck, which is phenotyping. But even I'm just going to address, I guess, that in two ways. Just from the phenotyping perspective, we sort of have, in my view, we're sort of solving the data acquisition bottleneck, right? Now, how do we make use of that data so that we can actually make selection? Implementing that into a breeding program, that's just sort of another question that we might answer. But the bottleneck at that point, I think, is going to be actually computational. It's going to be we're going to have there is some limitation in the amount of data that we can store, the amount of data that we can process at a given time, and the amount of data, in some cases, the amount of data necessary to train an AI model, for example. Okay, so data turnover, I think, it's going to have to ramp up in a significant way in these if we're to make effective use of this technology. Now, and then stepping out of the selection accuracy sort of component, I think one bottleneck that we need and we've always thought we know about, but it's too difficult to sort of address, I think. And when I've always had some interest in this, but I think it's a complicated genetics question, is how do we maintain genetic gain without sacrificing loss in additive genetic variance, right? So it's a difficult question. Is the breeding program inextricably requires screening or parsing through genetic variance, and so the total available variance has to go down. But how do we make that sustainable over time? And so there's questions there around that. There's new knowledge about our various genomes that we can potentially use to sort of to at least curve that a bit. So one of the things that I've always been interested in is how can we, for example, leverage areas of low recombination, and how do we target areas of low recombination so that we can design new ways of performing or designing crosses and new populations with the aim of uncovering new combinations from those regions.
That's fascinating conversation, isn't it?
Yeah.
I have another question from Perpetua here. Thanks for the wonderful presentation. And do you advise that we drop the old model of phenotyping?
That's a trick question. I think Well, I think it depends, right? There are things that we certainly can't observe, no matter how much we try, and there are things that can we can afford to continue measuring the way we do, but we have to be conscious of what we fail to select for if we continue with the same model. So we just need to, I guess, just be realistic with what it is that we are, that or very, I guess, very grounded in the goals of the prime breeding program, right? So that's really, I think that's really the advice or my view of it, right? If the breeding program wishes to extend beyond certain characteristics that require HTTP tools, then yeah, perhaps we need to be looking at ways of making selection in a different way. So I guess what I'm trying to say there is that it all really relates to the trait architecture. Okay, so if the trait architecture is made up of a set of conditions that we can largely modify using the same old methods, and the breeding program is making progress that way, I don't really see, there's maybe no real advantage from implementing HTTP, right? Because if the trait, if the implementation of HTTP allows you access to that, say, small amount of the genetic variance or the selection accuracy to gain a small edge in the selection accuracy, but it comes at a great cost in resources or even in other components of the equation, then probably not. But if the goal of the program has if the goal of the improvement of that part of a different trait, which is made up largely of small effect loci, and we have very a lot of trouble selecting for them, then this is really why we do this. Yeah.
I think it's a very, very good discussion, Valerio, because I find that the breeder's equation is a universal concept that it really help us visualize what are the trade-offs, and the resources aspect is not part of the equation as we have presented. It's time, it's efficiency, it's genetic variation, accuracy. But at the end of the day, the resources are the ones that determine how much you invest. If you invest all your money in high throughput phenotyping, then you don't have money to do other things that are as important. So
yeah, this is one thing that I've discussed with my New Zealand colleagues is, I mean, if you just use deterministic models, you can pump up genetic gain quite a bit. So to the point where you exceed, you can adjust the parameters in that equation to the point where you realistically, if you ran a very, very large breeding program, you could probably pump up genetic gain to a point. But when you include the economics of it, the cost is so overwhelming that it makes it not realistic at that point.
Right. Yes. And not only the cost, these are logistical aspects, because if you add to the high throughput phenomics all the high throughput genotyping that has to run in parallel, and merging that two aspects of the information and making it on a spreadsheet that the breeder takes and said, okay, we're going to select this, or your machine learning program will do that, hopefully. But then you have to recombine those and make it move. So question from Roshan: what's the strategy to combine HTTPs together for holistic selection gains?
Well, okay. So that's related to trait correlation, right? So it's an approach that I think, it's still sort of under development in my lab, and in other research groups as well. Is that, okay, if we have sufficient knowledge about, there's two ways we can approach this, right? We can do it with a sort of an unsupervised way where we just collect data we collect data on a particular using a particular sensor, and then we just simply try to brute force find correlation with some sort of so with the trait itself. Okay, so we use just basically we try to find, okay, what sort of amount of variation, amount of data, and which sensor specifically leads us to the highest level of association, and that's sort of the most straightforward way. Another way that we've thought about how to do this sort of is sort of a supervised method, where we take knowledge of QTLs and QTLs that are, as I said, not just the overall trait itself, but the individual components that make up this trait. And we try to model this, the set of dissected components in order to design a sensor system that would enable us to predict, to measure it with a sensor system. And so that sort of what I'm getting at. So it really requires some thinking from the breeder as to what do you think might be, what do you know about the trait, and what do you think might also be involved, so that you can put the right sensor fusion together. And then at that point it just becomes sort of a statistical exercise.
Mm-hmm. All right. Well, any other questions before we close this seminar? It's been very interesting conversation, Valerio. Thank you so much for your time, and I want to thank the people who attended the whole seminar series. This is the last seminar of this spring, and we would like to invite you for hopefully next fall. Usually the seminar runs between October and November. Sometimes we start at the end of September and finish early November. So thanks, Brandon and Thiago, for your help. We will share with the people that has registered for the seminar the recordings pretty soon, and then the whole seminar series will be available on YouTube later on. Okay, so thank you so much. Thanks, Valerio. Have a successful season. I know you are thinking about starting planting pretty soon. Let's stay in touch. I really like your concepts, and I encourage people to reach out to you in case they have more questions or want to learn more about your program. Thanks, everybody. Have a great season, a great summer, and look forward to seeing you again in the fall. All right. Thanks,
everybody. Bye. Thank you, Carlos.
Bye-bye.
Bye.